showed you this data. So, sorry, I guess I have to. All right. So you see that when I heat the blue curve, I get an extra 20 or 25 degrees beyond the ordinary TG before it becomes a liquid. But you're right. Once it's gone up to the supercooled liquid, it's forgotten all of the interesting structure that we provided by vapor deposition.
So don't put your cell phone in the sun, I guess.
Actually, if it's an OLED display, it's a bad idea to leave it in the sun for long periods of time. They are photochemically degraded by light over long periods of time. All right, let's take a question from the right-hand side. There you go.
Hi, thanks. How does this work change how you interpret experiments with like the confinement effect of glasses? And a lot of those are strongly dependent, obviously, on the substrate that you're confining with. And then I don't know, maybe I missed it, but I don't know if you talked about the substrate that you're doing the deposition on. And yeah, thanks.
Okay, great question. So if we often are looking at films that are between 50 nanometers and 1,000 nanometers, and in that range, the properties are independent of what the structure is. Okay, and that's consistent with our mechanism, which is all about templating from the top surface and not from the bottom surface. Now, certainly, if you get to really thin layers, you're going to be influenced by the structure of the substrate, and that's something that we're still working on understanding.
All right, let's go back over to the left for a final question. So I'm just curious, let's say I want to make a packing of oranges as dense as possible. Do I drop them one by one on whatever the flatbed truck? So does vapor deposition work for... granular materials in any kind of general packing, or does it only really work for molecular glasses?
Okay, that's a good question. Well, okay, I know people have done experiments on colloids, okay, sort of intermediate state, and they make a good glass, but it's not particularly any better than you would make by standard methods, and that's because colloids really don't have this. We've got 8 orders of magnitude of extra mobility at the free surface compared to the bulk. Maybe in colloids you got 1 order of magnitude or something. So you really don't have this kinetic advantage that we have with these organic systems. And so I think I have to think more about the granular problem.
Thank you. All right, well, let's thank our speaker again then.
All right, thank you, Paul. It's an honor to be here and to share with you some of the work that we're doing in my group in this field. So I'm sort of representing the biological perspective here. You'll see, though, that there really are a number of points of contact with probably all of the previous speakers to some extent, and certainly with Sharon's talk in terms of entropy as a driving force, essentially for self-assembly in cells. So if we think about biological systems, there's an enormous degree of richness to the sets of interactions. There are thousands of different types of molecules, biomolecules like protein and RNA and DNA that are interacting in a crowded environment. And so the interactions look much like you see in the simulation on the left. Those interactions, despite the enormous number of fluctuations, thermal and likely non-equilibrium fluctuations within the system, those interactions give rise to coherent assemblies, so coherent organization. Things like the migrating cell on the top, the leading edge of the migrating cell, or this neutrophil chasing a bacterium in the middle, or a frog oocyte or embryo that's undergoing gastrulation on the bottom. So I think it's fair to say that biological systems are the most richly textured form of matter in the whole universe. And if anyone has something more richly textured, I guess you'd have to define that, but then let me know. But I suspect we'd have to wait until we find an alien life form, and maybe it'll be more highly evolved or something. But that's a call to arms. I mean, in terms of physics, we suspect that there's going to be entirely new physics in biological systems by trying to study the principles that govern self-assembly. So what makes biological systems so interesting, one of the features that makes them so interesting is that there is a wide span of length scales involved and sort of hierarchical self-assembly. So you can think about, at the nanoscale, the building blocks, protein and RNA interactions, let's say, and somehow those interactions encode for organization within the cell, things like organelles, and then the function, the assembly and function of those things then dictate cell behaviors, tissue behaviors, and organism behaviors that ultimately can build soft, squishy robots like you and I. Now, what's interesting, I think, about this is that we actually know a fair amount, certainly not everything, but we know a fair amount about the physics that governs self-assembly at the nanometer scale, so this kind of smallest scale that I represent here of biological self-assembly. We can think about structural biology models, protein folding models. We have a pretty good conceptual framework for understanding things at that level. If we go up to the highest length scale, sort of what I'll call the macroscopic length scale, we can think about continuum mechanics models, the kind of cell vertex models that people think about in collective tissue behavior, or even, if you want to stretch it, you can say sort of physics-inspired behavioral models. So we can say something about what's happening at that scale. The length scale that I want to talk about as the focus of the discussion today is this mesoscale, at the sort of micron length scale, structures within living cells. And I would argue that we have very little understanding of the rules that govern assembly at that scale. So I'm going to try to tell you about some of the ways we're thinking about that. A lot of the work in my lab and in this field that I'm going to tell you about is inspired by soft condensed matter systems that are non-living, but have some features, qualitatively at least, that are similar to biological systems. They're squishy and they involve macromolecules, things like polymers. They can give rise to emergent behaviors, viscoelasticity, and so forth. So we're thinking a lot about the ways in what we learned in soft non-living matter and how we can take some of those insights into living cells and ask, can this give us new insights and new knowledge on how cells are organizing their contents? So if you think about what's happening inside living cells, that picture, the movie I showed you at the beginning, the simulation of all these molecules that are interacting, one of the ways, sort of high school biology textbook picture of how the cell organizes its contents is through organelles. And so many of you probably remember from high school biology the organelles with these funny names like endoplasmic reticulum, Golgi apparatus, and so forth. So essentially all the ones we learned about in high school biology are membrane bound, and they're to some extent vesicle-like. They're like little soap bubbles that compartmentalize the inside of that, that compartment from the outside, and so that kind of makes a lot of sense. But we think that that's probably just really the tip of the iceberg in terms of the way compartments form inside the cell and the way in which the cell organizes its contents. Turns out there's a whole broad class of membrane-less organelles, so bodies that are large-scale assemblies of thousands or millions of molecules that are forming coherent structures inside cells that play a broad range of biological roles. I'll tell you a little bit about these germline P granules in the top left here. There are things like processing bodies, signaling assemblies, and an array of these structures inside the nucleus, at some compartments of the nucleus. And in fact, I think it's probably fair to say that most of the bodies inside of cells, most of these kind of organelles that one sees, are in this category of membrane-less. So it's not, it's not sort of a small subgroup, but in fact, maybe most of the organization is mediated by these kinds of structures. And so I started thinking about these in the context of this problem, which these structures are called P granules, and they're implicated in specifying the lineage of the cell, so the fate of the cell, whether the cell is going to form a neuron or a gonad or something else. And so these structures are implicated in specifying the reproductive organs of the gonad of the C. elegans worm. And what's interesting is initially, after fertilization, you can see that they're distributed throughout the embryo, so these green puncta distributed throughout the embryo. But as the embryo starts to polarize and develops a distinct anterior versus posterior axis, these structures end up on one side. And when the embryo then divides in two, only one cell, the cell on the right, gets those structures, and that's the progenitor germ cell that goes on to make the reproductive organs in the worm. And so this process, we've learned a lot about how it works. It's mediated by gradients of molecules that are kind of morphogens that control the segregation of P granules into the embryo posterior. What's really fascinating about how this works is that these structures, these P granules, we discovered are liquid-like assemblies of RNA and protein. So despite the name of a granule, they're not little pebbles. They're actually condensed liquids. And you can see that in this movie where we apply shear stresses across the tissue in a very kind of low-budget rheology experiment. We just basically squash this tissue and do shear stresses. And what you can see is these P granules flow and drip and wet and coalesce with one another. And so this led to a model wherein the segregation of these P granules into the posterior end of the embryo is a kind of phase transition or a liquid-liquid phase separation process, just like what Sharon Glasser was telling us about in a non-biological context. So the idea is that the anterior-posterior axis of this embryo spans the two-phase boundary such that the posterior end of the embryo is in a two-phase region where these condensed liquid states of RNA and protein are stable. And since we first put forth this model, there's a lot of data supporting this, and it's a very rich problem, and there are many non-equilibrium features which I want to highlight. The cell is very much out of equilibrium. You can see already from the gradient that drives this assembly. So this idea of condensed liquid states inside the cytoplasm and phase transitions driving organization inside cells actually has a long history. We started to discover papers back in the literature as far back as 1899. Pioneering interdisciplinary biologist E.D. Wilson had some of these ideas about the physical nature of the cytoplasm, and he described the cytoplasm or the protoplasm as a liquid in the form of an emulsion consisting of these suspended droplets. In fact, we've known that biomolecules can undergo phase transitions for many decades, and a lot of work with structural biology where trying to induce proteins, for example, to crystallize such that you can do X-ray scattering. Phase transitions have also been suggested to potentially play a role in membrane organization, and so there's kind of hints of this in the literature. And it has started to become apparent that these kind of phase transitions are ubiquitous within the cytoplasm and the nucleoplasm, the contents of the cell. There's literally dozens of different, or really at this stage, hundreds of papers over the last several years that have come out suggesting that liquid-liquid phase separation is implicated in processes, ranging from mitosis to cell signaling, postsynaptic densities that mediate connectivity in neurons, and essentially, without exaggeration, I would say almost every biological process that's happening in a cell, there's some connection to liquid-liquid phase separation now that's been described. So we're really interested in this process, phase separation, and how it is occurring potentially to organize the contents of the nucleus. So the nucleus is where the genome sits, and so we're all familiar from 23andMe or something, with the idea that our genes are dictating phenotype. What's something that you may not appreciate is that every one of your cells, so this is actually a picture of my daughter when she was first born, every one of her cells has the same genetic code, the same sequence of base pairs that are supposed to dictate the phenotype. And yet she has dozens or hundreds of different types of cells that make up her body. So each one of those cells, its phenotype is supposed to be dictated by the genome, and yet you get totally different cell types with different shapes and different functions. So how does that work? Well, it turns out that it's becoming apparent that these kinds of condensed liquid states are like little dewdrops that are sitting on the grassy genomic landscape, and they're playing roles in mediating expression of genes. And we started to refer to this as a liquid epigenome. So there are many different types of these condensed liquid states which we think are doing this inside of cells. Structures like the nucleolus, which I'll tell you about, which controls ribosomal RNA biogenesis, so the protein translational machinery inside of a cell. Structures like nuclear speckles, which are regulating splicing, so how genes are being cut and cutting out introns, and so forth. And Cajal bodies, which are also playing roles in the regulation of expression of genes. So we started studying this with a postdoc in my lab, Steph Weber, who's now a faculty member at McGill University, and she was able to show that the nucleolus, so this structure that's inside the nucleus, the nomenclature is a bit unfortunate, so the nucleolus, and it assembles around these particular genomic loci that make the ribosomes. And she was able to show some evidence that the assembly of the structure seems to follow a phase boundary. This is we could change the concentration, and the higher the concentration, the more material condensed out of the nucleoplasm. We can also look at the dynamics of the assembly of these droplets and show that there was coarsening of nucleoli around those transcriptional sites that was consistent with sort of classic modeling approaches. So this is the Cahn-Hilliard equation, which is a kind of diffusion equation, except it's uphill diffusion. I was going to make a point about how entropy can only drive things downhill in Fixed law, but that may be totally wrong now that after Sharon's talks, I'll hold my tongue on that. But it's exciting to think about. But generally, one thinks about diffusion down concentration gradients driven by entropy. And so if you want something that's uphill diffusion, you would have to have some interaction, something beyond just entropy. And that's what's in that formalism. What's different here, though, is that we have the presence of RNA due to transcription at a particular genomic locus, and that's why the droplet in the middle wins out. Another system that we've been using to probe this idea that liquid-liquid phase separation is important in the nucleus, in particular with the structure of the nucleus, is a frog oocyte system. It's really powerful because it's a single cell. It just grows to be very large. It grows to be about a millimeter in size. So it's a single cell that you can hold in your hand, and it contains a nucleus that is similarly large. You can also see it with the naked eye. And inside that nucleus, there are hundreds of these bodies, hundreds of these nucleoli that are sitting there and can be mechanically probed and tested in other ways. And so this is actually one of the first systems we started studying. We showed after the pigranule story, and we showed that these nucleoli really have classic liquid features. This is a liquid-rich rupture after they start to fuse, and we managed to pull them apart. What's an interesting feature here is, again, there's signatures of non-equilibrium, the importance of non-equilibrium in cells, because if we remove ATP, which is sort of the molecular battery inside the cell, then the apparent viscosity of these structures goes up a lot. So they tend to fuse much more slowly, and their dynamics are slowed. So that's something that we're kind of actively studying. First graduate student in my lab, Marina Ferrick, who's now a postdoc at NIH, came at this problem and started thinking about, asked a very simple question, which I'm a big fan of asking very simple questions at the outset of a project. And so Marina asked the question, well, if these are all droplets, why aren't they all coalescing? What's keeping them from all forming one mega droplet, like in salad dressing as you shake up the bottle and come back? And so what Marina found was that inside this nucleus, there's a scaffold which is formed by an actin network, which is very interesting because it's a nuclear actin network. We're used to think of actin in the cytoplasm. And that actin network supports these droplets and keeps them from coalescing together. And what Marina found is that if she disrupts the actin network, these droplets actually do all coalesce. What we expected was going to happen was that they would all diffuse around and bump into each other, but what we found is they actually sediment under gravity to the bottom of this nucleus. And that was pretty surprising to us because I had actually been teaching in class how gravity is negligible in cells. And so Princeton didn't fire me, so that was good. But the calculation you want to do is you want to calculate this sedimentation length scale, which is essentially reflects the competition between entropy, which wants to mix up the system, so sort of captured by kT, and the gravitational force, mg, or here expressed in terms of a buoyant mass. And what's interesting here is that the cell has grown so large that it's actually exceeded this gravitational length scale. So that's like the equivalent of climbing a mountain in Bolivia and you can't breathe, right? The physics is the same. You sort of cross this length scale. And in fact, if you do this experiment, you wait overnight, you do find one single large droplet. And so this is the world record nucleolus. It's about 100 much bigger than most cells, so it's this giant thing. So the other thing that is really interesting about this work is that Marina found when she was doing these experiments and inducing this coalescence by disrupting the scaffold, she found that there are subcompartments of the nucleolus that actually themselves also coalesce during this large-scale nuclear coalescence. And so I'll show you this movie. The green shows an interior compartment that's characterized by the protein fibrillary, and the red is this outer compartment characterized by protein nucleophosmin. And what you can see is that as these nuclei start to coalesce, the inner core also coalesce. And one has a sense that what you're seeing here is a kind of liquid within a liquid, as you can see in these these higher resolution images on the right. And in fact, that's exactly what we think is going on. We were able to show that we could reconstitute this liquid within a liquid assembly in vitro with purified components. What's remarkable about that in vitro reconstitution is that the core shell architecture is maintained. So these green proteins, fibrillary in particular, is on the inside and nucleophosmin is on the outside. And that it's known from multi-phase liquid physics that the droplet with the higher surface tension with respect to the surrounding fluid will be engulfed by the other droplet to minimize that surface energy cost. So basically, if the high surface tension, surface energy, then if it's engulfed by the other droplet, that interface will go away and the energy will go away. And so we were able to do some surface contact angle measurements and surface tension measurements to show that was consistent. It was also consistent with computational work that we did together with Rohit Papu's group at Washington University in St. Louis. So we like thinking about this because there are dozens of different types of these structures, and often they do have subcompartments, or there seem to be multiple liquid-like phases, which includes things like the stress granules. So we're interested in this idea that the relative surface tensions again structures the contents of these other bodies. There are other there are other bodies that don't interact at all. So they're not engulfing one is not engulfing the other, but they're not fusing or coalescing or interacting at all. And so we think that reflects a high surface tension that would be associated with the interface between those two. Again, it's known that in multi-phase liquid systems you would have complete exclusion of those droplets. There are other biological systems where there's partial engulfment of the droplets, one partially within the other, which is associated with sort of relatively more equal surface tensions. And things like this beautiful image of this Cajal body and the attached structures called the synerphisome, which I always think is such a cute name, especially for something that looks like Mickey Mouse here. But this is a case we think of a partially engulfed liquid. So I haven't said much yet about the molecular driving forces that are at play here. It turns out that many of the proteins that are driving phase separation are referred to as intrinsically disordered proteins. So the proteins that are conformationally heterogeneous. So I think everybody knows that proteins are linear polymers. They're linear chains of amino acids. But we generally don't think of them as polymers because we think of them as having a compact three-dimensional shape that dictates their function. But it turns out that many proteins are actually conformationally heterogeneous. So rather than being folded into a single compact shape, they're actually sort of fluctuating around. And I'll show in this movie, this is a simulation of a protein protein like this, one of these intrinsically disordered proteins, or IDRs. These are closely related, by the way, to what are referred to as low complexity sequences or prion-like domains. And so we and others in the field over the last few years have been intensely focused on these IDR proteins that contain IDRs, because when you purify these proteins and look at them in vitro, what you find is that they readily phase separate into these condensed liquid states, which we think correspond to the same kind of molecular driving forces as the corresponding structure in the living cell. So this includes proteins like LAF1, which is one of a set of important IDR-containing proteins in P granules, these germline specifying structures that I told you about at the very beginning, and also with nucleoli. In the bottom two examples. And in this example, the yellow droplet, that's from a fungus. So these are just a few examples from our work, but there are really many dozens at this point of intrinsically disordered regions which are thought to drive phase separation. Another key molecular architecture of proteins that are driving phase separation is oligomerization. So a lot of the proteins, and we've just really started to fully appreciate this pretty recently, is that a lot of the proteins have they'll have these intrinsically disordered regions. So this is a linear representation of the domain structure of a protein. You'll see that blue intrinsically disordered region in the middle. On the left is what's commonly seen is that there's an oligomerization domain. So that could be a dimerization domain, so homodimerization, or the protein I already discussed, nucleophosphate, has a pentamerization domain.